Good evening, ladies and gentlemen, and really a warm welcome, particularly to guests and visitors. It's a great pleasure to introduce this evening's proceedings. My name is Klaus Dodds. I'm the Executive Dean of the School of Life Sciences and Environment. And one of the nicest things you do as Dean is introduce colleagues who are about to deliver their inaugural lecture. And for those of you who are not familiar with inaugural lectures, it's just worth stating that it really is a mark of considerable esteem to be promoted to professor. And then one of the ways in which we celebrate the election of someone who has been appointed professor is to give them an opportunity to profess on their subject. And so the inaugural is really a kind of an opportunity to showcase that expertise, that wisdom, but also fundamentally it's a celebration of someone's achievements. And I think, as this lecture will demonstrate, it can be an opportunity to reflect on all the help and support and guidance you've had along the way. So I think I'm very taken by the title of the lecture, and I think what it implies, because anybody who has made it to the rank of professor knows only too well that you really do need an awful lot of help and support from friends and colleagues and family along the way. So it's a great pleasure to introduce to you Professor Rafael Yáñez. Rafael has been at Royal Holloway for 15 years, and he is a member of the Department of Biological Sciences, which is an integral part of our school. In terms of research expertise, Rafael is really very well known for his work on spinal muscular atrophy, which is a muscle wasting disease, and which is something that affects children around the world. And one of the things I was struck by, Rafael, as I was doing a little bit of reading about the disease, is that it affects anywhere in between, I think, 1 in 6,000 to 10,000 children around the world, something like that. And then as a geographer, I also noted, I was reading an article that suggested it was more prevalent in Central and Eastern Europe compared to Western Europe, and that presumably there's a story to be told about the geographies of this particular disease. One of the things I think is incredibly important about Rafael's scientific work, and I think really epitomizes the school more generally, is a focus on really real-world research, where actually one is committing time, resource, energy to trying to find solutions to make a difference to people, environments, ecologies around the world. And I think for me that's one of the nicest things about being dean of this school, is the kind of impactfulness the colleagues, including our research students, have within the UK and beyond. I think it's also worth noting that Rafael has been the president of the British Society for Gene and Cell Therapy, and he has also been an extraordinary presence for this school, and indeed a champion, I would say, actually helping us to improve the student experience in our school and beyond. And I'm particularly indebted to him for his dedication around the work on mental health and how we support our students. So without further ado, I'm going to invite Rafael to deliver his inaugural lecture, and then after the lecture we will have a vote of thanks to be delivered by Dr. Andrew Porter from Imperial College. And Andrew, thank you in advance for agreeing to perform this really important task as well. So for now, Rafael, may I invite you to give your inaugural.
So for those of you who felt like running when Klaus said to describe my scientific expertise, the lecture tonight is more about photographs and art and my scientific career as opposed to what I have actually done. I will try to showcase that, but in a way that can be understood by anyone in the audience. And let's see how it works out. I've also tried to make the lecture interactive, so I'll ask you to join in the poll. So anyone who has a mobile gadget of any kind, please point it to the app that we are going to use, because you will have an opportunity to answer questions that I will be posing during the lecture, provided that everything goes well, because as life is busy, I think I finished putting everything together about 10 seconds before I was due to go and join the tour. So a catastrophe can only be round the corner if things turn ugly, but let's keep our fingers crossed. So obviously with that title, my purpose today is to thank the very many people who have helped me in my career over many years. And this is what I intend to cover today. Okay, so I intend to tell you a little bit about why I'm standing here, highs and lows of my career, how important it is to me to work with friends, plan B that I had to activate at some stage, how tough it is to get an academic job and survive it, the importance of rare diseases, that's going to be a theme pretty much across the whole of the talk, what we have done here at Royal Holloway in the last few years, and all with a bit of art in between. And as I often tell my students, when I'm lecturing, I'm performing. I'm as far as away from a clown, really, as you can imagine, but when I'm here, I'm performing. So I'm hoping that you will get some fun out of this. So how am I going to do this? I intend to cover a lot of ground. Obviously, it's not going to be a Dali type painting. It's going to be more like a mirror type painting, very little detail. If you want to know anything specific about something, just get back to me. You all have my email address. I said there will be photography here, and these are just some examples of photographs of mine taken over the years, and you are going to see quite a few of those. I also mentioned art. This is purportedly my first ever drawing at age 5. I must say that I haven't improved any, so what I do now is art management. So I get my ideas onto people and they do beautiful things for me. So you're going to see a few of those on the presentation, hopefully. You will have seen in the intro that sadly a number of friends have not made it to here, and for me this is a way to remember them. They are very dear friends, all of them, and this is a way to have them present here tonight. And I thought I would start at the beginning. Not the very beginning, as you can imagine, because I'm by here, I'm very well fed, but you know. And context. I was born in 1965, so 1965, Britain was, you know, paradise, if you want to, modern Britain in 1965. Spain, though, where I'm coming from, is a different picture. But not everything was lost because, hey, the Beatles came to Spain, believe it or not, in 1965. So, you know, it was not the end of everything. Now, obviously, my family has been very important to me all along, and this is a picture of the whole family way back, you know, under Franco. This must have been 1969. When my youngest sister was born. And that's what Spain looked like at the time, you know. But we moved on, and we developed nicely, I think, as a caring family, well away from the black and white image that both Spain and us had under Franco. My dad sadly didn't make it here, so this lecture is also particularly dedicated to him. He died back in 2010. And that's my mom. And my mom would have loved to be here, but she can't, so I'm sure that you will be okay, because I'm going to talk to my mom in Spanish for a minute. I'm not telling her any secrets. I'm going to provide you a translation of what I'm telling her, but right now I'm going to talk to my mom for a minute in Spanish, okay? So this is what I'm going to tell her. Mamá, sé que te hubiera gustado muchísimo estar aquí esta noche. Estás aquí esta noche, como siempre, como el resto de la familia, que ha venido inesperadamente tomando el vuelo de esta mañana. Yo sé que crees que no has hecho nada especial, pero lo has hecho, y yo siempre te estaré agradecido por tu apoyo, siempre tranquilo y callado, porque siempre sabías lo que estaba pasando, por acordarte y por tu cariño. Esta noche estás aquí más que nunca. Muchas gracias y te quiero, mamá. Okay. And you can relax because there won't be much Spanish on the rest of the presentation. And I'm going on to school, and this was my school in Madrid, throughout from reception all the way to joining university. And, you know, same as everyone, I think that you really never forget a good teacher. Alberto is actually here tonight. He's come all the way from Madrid. And I know that Alfonso would have come if he had been able. Sadly, he can't. So, Alfonso, si estás escuchando, un abrazo esta noche y que te recuperes pronto. They were my biology teachers in school. Alberto did the lecturing and Alfonso did the practicals. And very likely they are chiefly responsible for me being here tonight. But hey, what I really liked at school was basketball. And this was my first school team. I was age 10. I was going to ask you in one of the polls to guess who I was on that picture. Actually, sadly, the projection is darker than the screen, so it's very difficult to tell anything on the screen. So I can tell you that I'm this little runt down here. And I was little, really. I was always 1 year ahead of my age in school, and I was always the runt. That changed a little bit in later age, but you know, at that time, every year I was the little one. Anyway, basketball was really very important, and the friends I made have stayed with me. So every Christmas in Madrid, we have a Christmas dinner with friends with whom I used to play basketball. And this is a picture from the latest basketball Christmas dinner. Now, that's when we can be approaching disaster because I'm going to try to activate the poll. So let's see how it goes. So if I go to here, okay, and I open the poll, and if you have got the instructions, if you just go to vivox.app and use that code 119367891. And I think if I open the poll, you should see this question on the screen. Am I right? Can you see that on the app? Yeah? Cool. Well, answer the question. Don't be shy. Your life doesn't depend on this. You can just give an answer and don't take half an hour because they'll kick us out from here. I think you are being guided, perhaps misguided by the first responders, you know. Use the answer that you consider. But I'm going to count to 5 because we can't be here all night. So 5, 4, 3, 2, 1. And I think I can close the poll somewhere. Stop the poll. And that's what we have got for our responses. And you're right. We have celebrated 25 dinners because the 26th couldn't happen. We were all in lockdown. I couldn't travel to Madrid. And the 27th, I was in Madrid, but it wasn't advisable to get together into a group. So we decided to call it off. So we are all looking forward to this year's dinner. Fingers crossed. Cool. So far, so good. let's go back to the presentation. So the right answer, sorry, I should have shown you this slide. That was my cue to introduce the poll. Not very clever, and there you go. It was the 25th Christmas dinner. And hey, after school came uni, and it was a bit of a close shave, you know. I wasn't professorial at the time. This is actually my entry card for uni in Madrid, and you may see this was out of 10, particularly on the third exercise. I just made it. And the back, actually, lucky for me, is the marks that you got in school. So at the time, an average was done of what you scored in the exams and what you scored in school over the previous 4 years. And that was your final mark. So that means that my final mark, that calificación definitiva, final mark, was, is it 6.5? So I went to uni. You'll be glad to hear. But I must say that there were two crucial issues that prevented me from doing well at that exam. One is that our school, excellent as it was, really did not prepare us for those exams. I had not had a single exam in a school that covered the whole of the syllabus of a year. And for that entry exams, over 2 days, I had, I think, 8 exams covering the whole of the syllabus for each subject. Not 3 like here, 8. So that didn't bode well. But hey, there was also some maturity. Spain was hosting the World Cup in '82. For the first time ever, every game was on TV. Guess who watched every single game? So I don't remember anything of my preparation for uni, but I can tell you that Cameroon had a fantastic goalie and that their kit was quite remarkable and colorful. Yeah? But hey, there was an issue with maturity in there at the time, obviously. But I made it to uni, so I was a sort of a flatliner in school. I did okay, but nothing special. I just made it into uni. And that's the time scale and where I was. And you're going to see this slide increasing as we go on in the presentation. So I went to 2 unis in Spain at the time. A biological sciences degree was 5 years, so my first 3 years were in the Complutense, and the final 2 years were at the Autónoma in Madrid. But same as I tell my students all the time, you need something that gives you the edge when you finish your degree. And for me, the edge was doing internships in 2 different labs while I was doing my degree. So from the second year of my degree to the 5th year, I was an intern in 2 labs simultaneously, which is tricky, and I'll explain to you how we managed that. But that really is what gave me the edge. I finished with a decent degree, but I had a lot of lab experience by the time I finished my first degree. And that set me up straight into a scholarship. Because I wanted to do research. So I thought, let's define for you what science is, because we all know the word, right? What's science? But what's science for you? What's research for you? So for me, we are working at the edge, basically, at the limit of what we know and what we don't know. And the bulk of the scientists, of the researchers, are working there, you know, doing their little bit to push the boundary a little bit to gain knowledge. And truly only really exceptional individuals, not me, can push really hard and make a great difference to that boundary. The bulk of us are here. We are hardworking people, committed, interested, but pushing the boundary a little bit in a collective effort. And that is very important. This is exceptional. I'll tell you that out of all the people I'm going to mention here tonight, I only consider exceptional one person. I'm not going to tell you who that is for 2 reasons. 1, not to embarrass my friend, and 2, because that way every single one of my colleagues can go to bed tonight thinking, it's me. So I told you that I worked as an intern in 2 labs while I was doing my first degree, and that obviously is tricky. And the way we worked it out is because I wasn't on my own. I had a friend, Manuel Serrano, who was with me from school, and we worked together in the same 2 labs, so we could push the same project together, either one or the other, occasionally together. And we did that at an institute, which at the time was called the Centro de Investigaciones Biológicas. The Margarita Salas bit came later. And in fact, this, I'll show you, is this. So this was the building where I did my internship in this institute. They are no longer there. But funny enough, they have incorporated that statue to the logo for the institute. And I think it's important in the in the collective memory of the scientists in Spain. There is a big of meaning to this, which is effectively the students and the postdocs supporting the building. They are critical to the work we do. There is also something additional that I can't tell you in public. You can ask me later. But the result of that was my first paper. So the work was done while I was an undergraduate. It was published after I finished my degree. But hey, we got a paper out of that, which for me is not the paper in itself. It's because you learn the scientific method, you get the experience, and as I said, it really set me up very well for what was to come. So I said that there would be do's and don'ts in this lecture, particularly for people keen on a research career. Do get yourself an internship, get yourself a placement while you're an undergraduate, because it will make all the difference. Short training courses, both as an undergraduate and as a postgraduate, were also really important for me. Particularly important was an EMBO course that I did in Italy, where I learned to work with the engineered viruses that will become very important in my career. And it's also enjoyable. But, you know, I was a student at the time when I did one of these Fundación Juan Marc courses. This is César Milstein, a Nobel Prize winner, who is a co-discoverer of monoclonal antibodies. And this guy ran one of those courses when I was an undergraduate in Madrid. Now, I haven't told you, but the school in Madrid taught French. So I had 0 English when I went to uni, 0, right? On my second year at uni, someone put in front of me a pile of papers, scientific papers in English. One of my sisters, Pilar, who is here tonight, very kindly translated the whole of the paper. She was an architecture student. She was translating a biology paper. It is quite an undertaking. But what I mean to say is I had 0 English. I had to learn from there. And at the course run by César, I decided to be brave. I was going to ask a question, right? He is Spanish speaking. Every attendee was Spanish, but the course was run in English to introduce us to the language. So I decided to attempt to ask a question in English. And I did try, but my voice just would not come out. It never happened before. It never happened afterwards, but I tried to speak, and nothing would come out. He made a laugh of it. He gave me a glass of water, and we just moved on. The next day, I asked a question. When I was going to do it, he offered me some water. I said, I'm all right, and it did come out. Don't ask me what happened, but that obviously was quite embarrassing on the first day, but you just have to swallow it and move on. So do. Short courses introduce you to new technology, new people. Networking can be a real eye-opener and very important for career development. So, PhD project. I'm going to introduce you to a little bit of science. Now, you don't know this, but COVID means that everyone has a master's in biotechnology. You are just lacking the certificate. Because you know what a virus is, you know how they divide, you know how they mutate and create new strains, You know that you can use antigen tests or PCR tests to detect them. You don't need to know much more than that, right? So, my PhD project was with the African swine fever virus, which was an important economic problem in Spain at the time. This virus is a real killer of pigs. And in fact, it seems to be spreading in the continent at the moment, and there is some concern about the possibility of it coming over to the UK, where there has never been, in the wild, so to speak, African swine fever, even though there has always been research in preparation for a possible outcome. So, what did I do to start with for my project? Well, we were going to calculate the mutation frequency of the African swine fever virus. And for that, we just had to produce 17 virus samples in the lab. You know that you can get virus samples in the field, right, from different people. Same, but in the lab. We had to prepare virus DNA. You have heard of that all the time. We had to sequence the same region of the genome in all 17 samples, like is done in COVID these days, very straightforward, and compare those 17 sequences, looking for any differences between them. That was my initial PhD project. If I had to tell you this before COVID, we would have spent the night. But today, it's straightforward. You do have a master's in biotechnology. So now, how sequencing was done before, that's a different game, yeah? So this was a real lab in Madrid at the time, where we did the DNA sequencing. If I ask my students, how do we do DNA sequencing now? I'll be telling them, well, you put your DNA in an envelope and in the post. Seriously, that's how it's done. And you get the result by email. The game was different. At the time, we have to go in the lab, get these X-rays with the 4 letters of DNA, A, C, G, T, and read them manually. Literally from the bottom, in this case, A, T, T, A. So normally you would have a colleague next to you writing down what you said aloud. And then it had to be typed into the computer. That's how we did things, right? Slightly different now. So imagine my disappointment when, having done all that for over 2 years, having read 51,000 nucleotides, there was absolutely no change. For those of you who are familiar with Spanish culture generally, this is the equivalent of peanuts in Spain. It's Mafalda and Guille, her brother. So Guille is here working at the sequencers, then they are reading the sequence, Mafalda and Guille together, and then Guille realized that there are no changes, and he goes, sits down by the door and waits for Mom to come in. And so this is my art management. This was drawn by my sister Pilar, who is here tonight, following an idea of mine to encapsulate the disappointment. So, what else did I do at the time as an undergrad? Not an undergrad, a postgrad by this time, 2 years into my PhD. I was fortunate to have a scholarship that allowed me to travel to wherever for 3 months a year with a stipend to cover my expenses. And the first trip was to the UK. So, what did that happen? I should say I hardly spoke any English, literally at the time. So I was ready to board my plane, got to the airport. The airline had folded. I had to sit tight and wait for another plane to be available. When a plane became available, I came over and hey, train strike. I couldn't make it to Mike's lab where I was heading for that night. Luckily, his brother put me up. I got to his place probably at about 8 p.m. His brother Peter could not consider that I may not have had anything to eat for the day, so he just said, that's your bedroom. I've never been hungrier in my life. And when I went out the next morning thinking, this is like Madrid, every street corner will have a bar and I can have breakfast. I can guarantee you that a residential area of Ealing is not like that. It really is not like that. And language skills, right? And the Spaniards are going to pick up this quicker than English people. So you all, or many of you, have learned languages. Vocabulary is easy. When you don't know a word, you take the word in your own language, change the ending, and give it the pronunciation that you think is the one in the other language. So that was my game. I finally made it to Mike's lab, and there we were. Me speaking away in, I don't know, pidgin English probably, and Mike listening patiently until I finished. And when I finished, he said, "What's that?" And I pointed, and he said, right? Because in Spanish, you don't say computador. You can say computador, but actually you say ordenador. So I translated that ordinator. And, we managed to work well for those months. And in fact, Mike, we published a few papers as a result of that short summer internship. And we had a lovely time. But the language skills were moderate, to be optimistic, right? So again, do. This is really important. If you have the opportunity, take it, because it actually introduced me to technology that allowed us in the lab in Madrid to continue the project that I'm going to tell you about in a minute. So, part two, obviously after the disappointment, I wanted to do something different. The idea of undertaking another similar project was not on, so they gave me something different, more sequencing, but we were sequencing parts of the viral genome that had not been sequenced before. So we described a few genes, so eventually my time came to write my thesis, so that's me in my bedroom writing my thesis, which was that lovely green book, and more art. My, at this stage, still girlfriend encapsulated my PhD onto this drawing, which says on the left a happy piggy, and on the right an unhappy virus-infected piggy. In terms of condensing the message of the thesis, was not quite right, but it was good fun. I should say that she did this for fun and had no idea that I actually put it on my thesis. So it's there immortalized on the second page of the thesis. So at the time in Madrid, you still have to do it. In Madrid, a viva involves a public presentation, like here. I could be doing my PhD thesis with a tribunal of 5 people, and you can see that I was using slides that young people probably won't even have seen. And this is a picture of me with my 2 supervisors, Eladio, who sadly passed away some time back, is one of those friends, and Esteban Domingo, who is alive and kicking and very active because he's a mega expert in virus evolution, and his expertise has been called on all the time throughout the pandemic. So, we went on from that with all the expertise that I had gained, and what I was proposed to was, let's go and complete the full sequencing of the virus. This is a big virus compared to COVID-19. So we did it, but for me it wasn't lab work, it was computer work. I spent countless hours working in an early Mac, an SE20, and in this PC. In the PC we put together all the little bits of sequence to generate the longer sequences, and in the Mac we analyzed them. But at that time we were still very much paper-based. So here you have all the binders that I went through to analyze every gene in the virus. So, another question for you. Well, let's see. Okay, I think I have to go on to open the second poll. So how many binders did I pour over when analyzing the complete sequence of the African swine fever virus? They are all there in the lab. So again, your life doesn't depend on this. Make an educated guess, and let's move on. So, how many? So, 5, 4, 3, 2, 1, and I'll close it. And there you go. Well, the answer was actually 310. The virus turned out to have about half as many genes, but we had to cover the eventuality that some very small ones were possible genes. Altogether, 310. I had a really fun, 18 months, maybe 2 years doing that. Every morning, I would sit at that desk, take 5 of the folders, and analyze them for the day. And the next day, same thing, until it was 310. Oops, sorry, that was the wrong one. I should be here. So we produce a paper. We actually produced quite a few papers out of that, but this was the main one, the complete sequence of the virus. To be fair, about half of it had been determined in advance by many people in the big lab in Madrid where I was working. What we did was to complete it. So to sequence the other half and analyze the whole thing. That we did, and that was a very big paper. But I wasn't on my own. I was sort of co-directing that with Javier, who again is a close friend, and is a bit of a theme in my career, having the blessing of being able to work with friends very closely and doing really well in that respect. So this is an important paper in this field, because with all the sequencing available, we know all the genes in the virus, and loads of different studies can be carried out. So for that field, this was a very important paper. But overall, in science, it's not a huge paper. So it's not in one of the mega journals that you really want to publish in and we'll touch on a little later. It's not Nature, Science, Cell, or the Nature family journals. It's just a virology. But it's actually a great paper with a massive amount of information and really important in that field. And I'm actually very proud of that paper. So, hours spent in the lab every day. And those guys become your friends. We were messing around here, waiting for a group photo. Someone said, everyone turn to the right. So there you go, everyone messing around. Or traveling together. I learned to ski with a group of friends from the lab. And there is me about 3 seconds before I fell flat. But hey, the picture, if I don't tell you that, the picture looks good. But science can be a pig as well. So you have already seen this picture. How do you sequence today? Effectively in sequencing factories. Yeah? The job is very different now. And another question for you. So if we were to do that today, how long would it take us to sequence the whole virus? And again, fairly quickly, so, 5, 4, 3, 2, 1, and I'll close the poll. And yeah, you're right. It will take what took, probably if we take the whole viral genome, it would have taken 4 people, maybe 8 years, working full time to do this, a significant amount of money. If you do it today, 1 person, 2 days, £100. Science can be a real pig, obviously. So, let's move on. In Spain at the time, you had to do a national service. So I did a national service, and mine was not military. I bailed out and I said I want to do the social service. So I worked in a children's residency. So these guys were either neglected or abused children who couldn't stay at home and instead were hosted with nuns. They lived very nicely with 1 nun in a small group in a sort of flat-like room within the residency, and it was a really nice environment. But my afternoons for a year was running a bus route, collecting them from local schools in the north of Madrid, taking them back to the residency and doing their homework with them. Great fun. They really were hard workers at their homework, haha. So they were interesting afternoons. Like, we had a lovely time over there, and we engaged very much, and we did lots of things together. And it was obviously a massive change from the day job, which was poring over those binders with the results of the sequencing of the virus. So back to this, my perception of my career, where we have been through school, through uni, through PhD. I was now a postdoc, and really it was my time to move on. I shouldn't stay there. And so what next? Well, I can tell you that the destination was set, because when I came for the first time in '89, I met someone who 5 years later became my wife, and can normally be found somewhere around vegetation. It can be trees, it can be a garden, it can be an allotment, you know, it's that kind of thing. It's a family thing. So, destination was set, London. But the topic I could choose. So what? More of the same? I had great training. I could have joined at the time the starting Human Genome Project and perhaps done well. Or something different. I had done loads of sequencing. I was bored silly with it, and I could go on and work on another thing. So, hey, now a question for you. So if I hide this and move on to this one is, yeah, what would you have done? So no one is interested in the Human Genome Project, you know. Our life depends on it. Okay, so I'll stop it in 5. So 5, 4, 3, 2, 1. And well, I must say that you have chosen to do very much what I did. I decided to move on. I had become interested in the use of gene therapy, which at the time was an idea. You couldn't do gene therapy. We believed that perhaps using genes and stem cells, we could treat people. But there was no demonstration. We were effectively making a leap of faith. And you have a definition of gene therapy there, and you have a karyotype here on the bottom corner, and a particular gene labeled in green on the chromosomes, one of maybe 25,000 genes in the genome. So, when you have a faulty gene, you can have a rare disease, and I'll show you why. But there is actually a definition for a rare disease. A disease is rare in Europe, and still in the UK, if fewer than 1 in 2,000 people are affected. But there are probably more than 10,000 rare diseases. So take them all together, and they affect about 7% of people, and they take a disproportional part of the health budget. In addition, most affect children, and many of those sadly die before their fifth birthday. And most of those rare diseases are genetic, so potentially we could treat them with this. So let me refresh your memory about what genes are, really very, very thinly. Genes basically store the information to make a protein. They are a segment of the chromosome, so you have a chromosome here, I'm sure you are familiar with the outline, with a loop coming out of it which encloses a gene. If the gene is wild type, then you get the normal protein. If there is a mutation in it, like this green blob, you have an abnormal protein or perhaps no protein. And that's a list of rare diseases. There is a list of rare diseases. It's phenomenally long, so again, question for you. So if I hide this, and so this one, if you were to read aloud the list of rare diseases, just to read it, how long would it take you? So again, make an educated guess fairly quickly. And some of you I know have attended at Rare Disease Days, so you should remember the answer. But so 5, 4, 3, 2, and 1, and I'll stop the poll. And the correct answer beats me why it's shown here, because obviously it's giving the game away, but it's 18 hours. It would take you, just to read the list, you would take 18 hours. People can spend their lives working on a particular disease, trying to understand it, never getting anywhere near a treatment. So imagine the task ahead, how important this is. So it was my time to move on, and from Madrid I had agreed to go to King's College at Guy's Hospital with Professor George Dixon. I applied for loads of different scholarships with him and fellowships, and went back to Madrid to finish my national service, where I got a handwritten letter from George saying, I moved a few miles across London to Royal Holloway. So in my next visit to London, I went to see where that Royal Holloway was. And having visited it with my wife, we decided that that really, really was not practical. So I wasn't going to go to Royal Holloway. So I looked for an alternative job, and I ended up working for MRC at the Clinical Sciences Centre. This centre was renamed and is currently named the London Institute of Medical Sciences. But in my time, when I joined, it was the MRC Clinical Sciences Centre. And what we did there was gene targeting that would become genome editing. And Andy, who is actually going to be doing the vote of thanks at the end of this lecture, was the head of the lab. And Andy was really a pioneer, because he at the time was proposing the repair of the genome as a therapeutic avenue, well before it was really possible in practice. And again, just what we were proposing is developing some sort of vector to carry our repaired DNA, correct a mutation in the genome, and effectively repair the fold. That's what we were trying to do. And obviously, it was very difficult, but we did try. So after several years working with Andy, I did my best to become an independent investigator. So what you need to do for that is you need to write loads of grant proposals and scholarship applications to every single place to see whether someone will give you one, which includes your salary, and you become an independent investigator with your own line of research. That is actually very difficult. It's a tough time writing those proposals and grant applications. So there you are at the computer working all the time, and sometimes you really receive some unsavory replies. I'm actually highlighting, obviously many are going to say, no, that's not unsavory, that's an expectation. It's what they say when they give you the reply. And I'm actually using an example from a paper as opposed to a grant, because the grant would have taken longer to explain. So this is from a paper. A reviewer of a paper of mine in the past, having sent a typed review to the journal, felt like going back to the journal with a handwritten fax to just hammer it completely, saying that we had shot ourselves in the foot with our amateurish analysis. So people, on occasion, under the peer review system, feel that they can say this anonymously. So you really need to toughen up, because unfortunately things are better now, but if the editor of the journal or the person who manages the grant program doesn't call this out, then you can still get a really unpleasant reply. And it's life. You just have to bite the bullet. That's what there is. But I moved on to a different institution, the UCL Institute of Child Health, where I carried on trying to do gene repair and writing away proposals. And we started doing something else. We started engineering the HIV virus to deliver our cassettes of interest. So we were creating effectively engineered viruses at ICH. But things, weren't going great. So another poll for you. And this one is on, so I hide this, and that's your next poll. How many proposals did I put in when I tried? And I should say every proposal is a very significant amount of time. It can take weeks to months. So it's a significant effort. So again, just to cut it short, 5, 4, 3, 2, 1, and I will close the poll. And you're right, I wrote 22 proposals over a few years in the late 1990s and the early 2000s, which is a good attempt, let's say. So I should say, actually, in fairness, that one was funded by a program in Madrid, which is very prestigious, the Ramón y Cajal program, to get people back to Spain after extended periods abroad. And that was the first call of that program. In Spanish, as a joke, because so many people with loads of experience abroad applied, is called like the best jamón serrano, the best ham. Those are the Cajales de pata negra, so the black leg ham Cajales. So I got one of those, but I decided that I really didn't trust the system. While I didn't have anything set here, I felt more confident staying with my family in the UK than going to Spain for 5 years and then what? And, again, that's what if I could have come back. But I did need a plan B. So what was my plan B? I needed a job. At that time, my job at ICH had finished. I actually ended up on the dole. It's a long story, but that's what happened. So I took the first job that was offered to me. I went to Barts and I worked on prostate cancer with Yong Ji Lu, who is with us tonight here. Very kindly hosted me, and again, we published a good handful of papers. I've never been a slacker in that sense. But the plan B is unknown to them. I started an environmental degree with the Open University, so all my spare time was going on that. And it was a bit of a killer, so I really sympathize when we have students who are doing a degree and they are working at the same time, because it's a real killer to do both things simultaneously. But hey, the work that I had started at ICH had continued in the background with me pushing in after hours, and we were able to demonstrate that these engineered HIV viruses were very good to produce the protein that we wanted in the cells. So that actually became one of these really big papers, an HR medicine paper, well after I had left ICH. And again, in consonance with the theme, I worked very closely with many friends, including Steve Howe, who was really kind, helping me to keep the work going, because he was still in that lab in ICH while I had moved on. I can't stress enough how important a paper in a top journal is for a career. I wouldn't be here tonight if I didn't have this paper. So I always tell my students, if you ever have a chance of publishing a paper in a top journal, throw everything you have at it, everything. This took me a year. Once we have the work done, it took me a year for the journal to accept it. 3 full submissions, every time turned down, and we kept coming back until we got it in. So it makes a massive difference. Because I had left that lab, it's well possible that my contribution to the paper may have diluted, and I had ended up somewhere in the middle of that paper, or maybe even the paper would have not been produced if no one was leading it. With the help of Steve, I was able to push that, even though I was by then in a second lab after ICH, and we published this. So one of those do's, make sure that if you have the chance, take it. Throw everything you have at it, because it will make all the difference to your career. The second do is stand up and don't let people take what you have worked for, because if you don't stand up, they'll push you over and they'll take the credit, and I wouldn't be here standing today. So another one for students and perhaps more junior people. You really need to stand up and defend what is yours, because otherwise someone is going to take the credit and you're going to miss out. And people will respect you if you stand up and defend what is yours. They won't push you anymore. If they can push you once, they'll push you all your life. If they can't push you, then they won't. So, in addition, that work was it was an HR medicine paper because it was very much against what was believed in the field, that those engineered viruses were not good at gene expression. And we were able to look at similar data, and actually they were. And we did the right experiments to demonstrate that they were. So it's really important to look at your data by yourself. Don't follow the peer pressure, and don't do what everyone is doing, right? Really important in your career. So, my perception, we are picking up, there is a chance there. I'm still somewhere in the cold. I was at King's working with Mike Antonio, who is here tonight again, on another theme, but this was back in gene therapy, while the prostate cancer work was not gene therapy. And King's was a really supportive environment. I had loads of colleagues who helped there and were interested, but there were no job prospects. The institution was not interested in that work. So I really needed to move on. But while I was there, I got a small grant from the Friends of Guy's Hospital, £4,000 for a scientific grant. It's peanuts. But that allowed us to produce a whole set of papers with collaborators in King's, which again became really important. So I always tell people, don't dismiss small grants. You can do a lot with the small grants. You really want the big ones, but you can do a lot with the small grants, so don't be dismissive. And I did get a larger grant with Spanish colleagues because I kept the interactions in Spain, which again was very important for me to get the academic job, which is what came next, the Holy Grail, an academic job at Royal Holloway, believe it or not, right? That place where I didn't want to go. And in fact, I actually lived further than where I lived to start with the first time I came to London. Anyway, we continued working with those engineered viruses and with the DNA repair. That's what the lab has done for the last 15 years. And, finally, I had got over the line. I had an academic job, which is what the academic job gives you is security. You are no longer on fixed-term contracts lasting 2 years, 3 years, on someone else's grant. You now have a salary, you can apply in your own right and develop the kind of research lines that you want. That's why it is so important. But it's also, as you have seen, really tough to get there. And why Royal Holloway? Well, because as I told you, many of those rare diseases are inherited. We could use genetic and stem cell therapies, and that's a strength of this institution when I came, and continues being a strength. So in the lab, we have worked on a variety of diseases. Klaus has already alluded to spinal muscular atrophy. We are doing a lot of ataxia telangiectasia work currently, and we have worked on other disease models on occasion with colleagues. I ended up working with George Dixon. He didn't hate me after I actually didn't come to join him as a postdoc. In fact, he was extremely supportive. He's always been in my time here, and probably he was instrumental to bring me here. And later with Linda Popper, but also other diseases. I'm not going to go into technicalities, but just so you know. And the work really is done by these guys, those guys that support the building over the years. I'm just clowning around. This is at graduation, the only time when we use the academic gowns, so I clown around, but that's what I do all year round. They are the ones doing the work, really. And that's their names. In fact, they are color-coded, male, female, so you'll see that through the lab, we have had more females than males being trained, which I'm quite proud of. And hey, back to that, they are the guys who support the building. They are the ones who do the work. As well as our funders, because research is really expensive, so without the funders, we would not be able to do this, just fair and square. And in the context of that, my sport has changed. I haven't really played proper basketball for a long time. I do cycle occasionally, and I've done cycling to support some of the charities that have funded the lab, particularly SMA UK and Action for 18, the two main charities funding the lab. And we have a fantastic network of collaborators all over the world. Not everyone is here because it would be impossible to read this. And we have done a lot of work in the context of 6 different European consortia, which have been really, really important for our research. So that's how important the European link is for us. But again, apart from the students and the postdocs, it's really important to have the technical support. The technicians provide continuity. The students and postdocs move on after a set period. So it's really important to have tech and admin people who provide continuity and allow us to do the work we do. And some of these guys are from here or from before. And we do a lot of outreach work. So you're going to see in a few slides what we do for Rare Disease Day. Rare Disease Day is set on the last day of February every year, because on a leap year is a rare day. That was the reason to choose the last day of February. So we welcome secondary school students who come to our Rare Disease Day. They listen to lectures from experts. They do hands-on work in the lab. They attend a stakeholder exhibition with lots of partners in this space, and they interact a lot with our undergraduate and postgraduate students in the course of the day. And they love it. We have had schools coming back for the 12 years we have run the event. And these pictures are our undergraduate and postgraduate students who effectively ran the day for us. And for me, it encapsulates what Royal Holloway provides, because everyone from the leading team, either the principal or a vice principal, would welcome the visitors. We will do the stuff in the labs. The postgrads will do the more technical stuff. The undergrads will provide all the support and run a number of the activities. So everyone in the community gets involved in this. And it's really appreciated by the schools. And in fact, some of our visitors tonight are stakeholders who exhibit at our Rare Disease Day. And hey, COVID is strict, and we couldn't have a presential event. So what we did in 2020 was send the resources and run a virtual event. So because we couldn't use the labs, we were in lockdown, only key workers could go to work, we were lucky to be able to take over one of the huge practical labs and effectively create a chain to put together the resources that we were going to send to 200 students who in the end were actually at home, not in a school. And there it is, us putting together boxes. You would not believe how long it takes to put together 200 cardboard boxes. I think I did 180 before I was, and that's the product. We shipped 200 boxes to about 20 schools who then provided them to the kids so they could do Rare Disease Day at home. And I'm really proud of that. Again, it was with the help of postgraduate students and undergraduate students that we managed to do that. Now, you have seen a lot of my career. I'm watching the clock. I'll try to wrap up relatively quickly. I think there is something very important that I want to cover. Is if you ask me, do I have any regrets? And I would say, one. Huge. Which is, at some stage, I put myself in a position that I could have a mental health issue. And I was partly responsible for that. And I regret that massively. So let me tell you what happened. But just in case anyone is affected, those codes would let you visit either mine or an equivalent Spanish charity, or you can reach out to me. So if you hear of an academic, I guess that something like this comes to mind. You know, someone with their feet up. Well, this was my workload in late 2019. The top set were my jobs here at this institution, and lower are the ones that I was doing for external engagement. And obviously you read the list and you say, well, I mean, that was bound to happen, right? Why do you become involved in all this? But I didn't have much of a choice for most of this. In fact, I did say I did not want some of them, and I still got them. And these are my fault, but sometimes you can't really plan. Things develop in a way that you end up doing something that is not necessarily something you have planned. Obviously, it's a pretty massive workload altogether. But our academic life pushes us to do this, because if you look at the promotion papers for us, for any advancement in the career, you are expected to do everything you did before and the new stuff. You never untick a box. You always take on more work. That's how the career is set up. This is the paperwork for promotion at professorial level here. And you see the sentence at the top. You have to do everything that you did before, as well as new stuff. So it's a bit of impossibility, isn't it? So what did I notice in late 2019? Well, I was really getting really stressed. And I went to a conference, and I shouldn't have gone. Became more stressed during the trip. I started actually avoiding colleagues at the conference. I didn't know what was going on other than I was really stressed. I couldn't prepare the presentation I was due to give, and 5 minutes before I was due to do it, I pulled out. I spoke to the organizer and I said, I can't do this, sorry. I avoided colleagues for the rest of the conference. I actually wondered whether to return to London altogether. Why? What for? But I did board the plane. And on my way back, it really felt like that. Everything I've done is absolutely wrong. And in the context of Brexit, I believe that I brought the college into disrepute. I know, it's not realistic, but that's what was in my mind. So I actually thought that my behavior would trigger riots. And as we were landing, that's what I expected to see. As we broke through the clouds, I was expecting to see fires in London, which weren't there. At home, I believed that my family was going to be the target of attack. And as you can imagine, I was creating havoc around me, both at home and at work. And obviously, I had to go away and deal with it. So, diagnosis by a psychiatrist, acute anxiety. But problems don't come on their own. All this came along as well. So, I believed things that weren't real. I could not leave my bedroom for a few days. I was dying every night. My wife can tell you that I was dying every night. Completely lost my self-confidence and had a massive weight loss in just a few weeks. So, it's serious stuff. So that's what it felt at the time, that really my career had gone to pot completely. I wasn't going to be able, not go back here, just actually go and buy something in the shops. But with, what triggered that? I've actually done a lot of thinking in the last 3 years, as you can imagine. So what triggered that to me? That workload is manageable if everything goes well. But you know full well that that's not the case. Things are going to go to pot at some stage. So that's what started happening. I lost key people in the lab, and I was struggling to do the direct supervision of students. The time and the detail that was required, I hadn't done for a long time, and it was a struggle, a big struggle. This was a job that I very much loved, actually, because it allowed me to work with Michelle and Elaine, who are really competent people, and we had a fantastic personal interaction. But that job was going to be taken away from me. I thought we had done a fantastic job for the 10 years. I really enjoyed that interaction. And while a reduction in workload would be beneficial, I was really sorry to lose that particular job as a result. I still am the director of this center, and I feel I'm not doing anything. I don't have time to do anything. And that really I wasn't happy with. And externally, we were in a financial crisis. I was the chair of trustees of a charity, and I was really concerned with the charity folding and having to make people redundant, really concerned with that. And the workload as editor of the journal just kept increasing. And I just could not handle it. Literally, by the end, the feeling is I cannot solve anything. No matter how simple it is, I can't. This is in the context of Brexit, some more art. This is a painting by my son as part of his A-level art. And this is what we Europeans felt after the Brexit referendum. We were labeled as migrants. It was a really unpleasant climate. And having worked in this country, in my case at the time, this was 2016, so it was 21 years. We got a label, a stamp on ourselves, and it really became unpleasant. And obviously this didn't help. And that was the overall background of all this. So, what happened? Obviously, I had to go away. I had to have medication and sort myself out. But there are always positives. You know, when something happens, something is going positive. And, well, my family here in Spain obviously rallied around me. My colleagues took my workload away and delivered my duties, and the NHS actually gave me great support. But there are 3 people who actually did massively at that time and before. Versha, who is here tonight, led one of the projects in the lab, the ataxia telangiectasia project. In a way, it's her baby. And she produced records and experiments in such a way that when her time to move on came, everything was perfect for us to understand. I could come back to those data a year later and understand everything and make myself up to speed on the project. And I will never be thankful enough, Versha, for that. But Versha had to move on. And Ellie, who at the time was a PhD student, took the brunt of it. She had to effectively run the lab, support a more junior PhD student, support our final year our research students doing their projects in the lab, and she took a massive amount of work, being herself a PhD student and part-time research assistant. So Ellie got through a massive workload herself and did fantastically well. And again, I could not thank her enough. And finally, Sahar, who is here recently, 3 months ago, undergoing her online Viva, just put her head down, carried on working, and did the best she could with the support of the other guys. So I'm incredibly proud of these 3, because they did really, really well. When I was away, I just had to step back, get better, and they had to get on with it. So 3 of you, you know, I'm really so proud of you. And in my perception, my career was coming back up a little bit. So this is kind of the final run of this. Why are these diseases important in addition? Because we understand what we have to do to treat them. A different matter is whether we can do it, but we understand what we have to do. And this technology has actually been developed on rare diseases, but is now being applied to more common diseases. So that makes it additionally important. Some marketed products of gene therapy. Remember when I came in the field, 95, this was a dream. Now we have marketed products, which can be either chemically synthesized nucleic acids, engineered viruses, or genetically modified cells. The slight problem is the price. And as the dollar, pound, and euro are more or less at parity, you can see the list prices for those drugs. They are phenomenal. And that is a really important problem in the field and needs to be dealt with. What difference does one of those drugs make? This is a drawing of my daughter depicting a child affected by spinal muscular atrophy, one of the ones treated earliest with Spinraza, a chemically synthesized nucleic acid. These kids, all of them, would die before age 2 if they have the most severe type of spinal muscular atrophy. But Cameron Harding, got the early treatment and was able to meet developmental milestones like sitting up unaided and standing up that were literally a dream for this. So this is the kind of effect that gene therapies can have on people. But not just that, because most of you, if not everyone in this room, would have been vaccinated with either an RNA vaccine, an mRNA vaccine, or an engineered viral vector. So mRNA vaccines, the BioNTech Pfizer or the Moderna, or an engineered viral vector, the AstraZeneca. So gene therapy technology allowed us to move on from the COVID-19 pandemic. These are squarely gene therapy technologies. So it's no longer just important for the rare diseases. Now this technology is mainstream and has massive economic value. But we are much better at diagnosing by genomics than at treating. I've already shown you those sequencing factories. If a child is born in England today, the child will be screened for 9 rare diseases. And I've told you that there are literally thousands and thousands. And that is, in a way, availability of treatment and cost. But for diagnosis, you can diagnose so many more diseases today. But what we need is therapies. When a child or an adult has a disease, what we need is to invest in the development of treatments, invest heavily. And that is not being done. Currently, the funding is going to sequencing technology. So we diagnose people, but once we have diagnosed them, we hardly have anything to offer them. And for about half the people who enter the genomics diagnosis stream, we still can't tell them what they have. So 6,000 children every year are born in the UK for which genomics cannot provide a diagnosis. We don't know enough for that. And for the ones who get a diagnosis, for the great majority, we have nothing to offer them. So there is a genomic medicine revolution going on, and I always tell my students, join it in the lab or in the clinic, because this is now in the clinic, or if you hate the lab and don't like the clinic, there are data to analyze, to your heart's content for your life. So just do it. Very quickly, I've already mentioned the British Society for Gene and Cell Therapy. I love working with these guys. I've done it from 2010. If nothing happens, I'll step down as president in 2025, 15 years of my life, and I'll love every minute. And again, with the Genetic Alliance UK, I was with them for 5 years at a very hard time for me and for them. I'm delighted to say that they pulled through and they continue doing the lobbying work on behalf of the rare disease community that they have always done. And the journal keeps going strong. I'm delighted that I switched from an editor-in-chief to an international team, one editor per continent, and as an excellent balance, actually, male-female, which there wasn't there before, and the same with the editorial board of the journal. And I'm delighted to see that they continue going on strongly. And just to finish, a little bit more art. So I'm going to tell you how the logo for my lab came about. So I wanted a logo for the lab. So I asked one of our undergraduate students who was graduating, whom I knew was a fantastic artist, to come up with a drawing. And that's what she did. Sadly, her dad was going down with MND. And that's what inspired her to produce this, which is a rare disease dancer. And you can just about see that there is an hour clock making up her body. This was too complicated for a logo. So my daughter, who is very artistic as well, Andrea, stylized that logo and made it possible to incorporate it into the lab's logo. And the last thing I want to show you is some living art, if this works. This is the north facade of Founders, illuminated in the colors of Rare Disease Day. So on Rare Disease Day, many iconic buildings all over the world are illuminated in the colors of Rare Disease Day. And the last day of February this year, we did that as well. So this is a picture from that day. If things work out, we actually have a live stream of what Founders should be like. If I can come out of here and don't end the show, so pointer options, arrow options, visible, and if it works, 2 phenomenal student ambassadors will have created a live stream of Founders illuminated tonight. So Founders is illuminated tonight, and there it is. It's not a very clear picture, but we have 2 ambassadors walking across Founders. So before you leave tonight, if you want to see it illuminated in the colors of Rare Disease Day, it's on. Yeah, courtesy of college. So it's obviously nicer than it looks on the screen. You have seen the picture. But it's there for you to go and enjoy tonight. And I'm coming back to finish. So, oops. I've actually met really inspirational people affected by rare diseases. Not inspirational because they have a rare disease, but because of what they achieve despite the fact that they have a rare disease. And all those mentors I've mentioned, my school teachers, the people who supervised me as an undergraduate student, my PhD supervisors, people who supervised me on short-term internships abroad while I was a PhD student, and people who supervised me, my line managers in the UK, And George Dixon at this institution, who was not formally a line manager, was incredibly supportive throughout my career here. And if you think this information would be helpful to other people, well, let them know that this would be available as a recording shortly online. And my final thanks, well, last but final, obviously, is to the family. My wife, Doreen, has been with me throughout highs and lows, and some of the lows have been fairly dramatic, as you have seen. And my kids have been at the receiving end of short-term care, as well as loads of parental advice that my students may follow, but my kids don't seem to follow. Right, but obviously, thank you to them, same as the rest of the family, as I did before. And finally, thank you, because I know you could be doing lots of things rather than listening to me here, so thanks for your time. And we will finish now with the vote of thanks a few minutes from Andy Porter, and thanks for your time.
I think Rafael, that applause gives you a sense of really what a barnstorming performance that was. And thank you for your honesty, I think your humility, and also for really giving us all such a positive note to end on. And I say that as somebody who's just had his 4th dosage of vaccination, which I'm also very grateful to the NHS for and all the work that goes behind it. But one of the nicest things we also do in an inaugural is invite someone who often has a very special role in the life and the development of a colleague who's been appointed a professor. And I think you've got a pretty strong sense of how important this colleague is. And it's a great pleasure to invite Dr. Andy Porter from Imperial College to give formally the vote of thanks. Thank you, Andy.
Thank you very much. Well, Rafa, well, my job is just to say thank you to people. But first of all, of course, Rafa, where are you, Rafa? I've lost you. I mean, what a talk. It was informative, it was amusing, it was moving, and I'm not sure I can really follow it very well, but thank you very much for that. And also, I need to thank you for the part you've played in my life as well. I mean, you mentioned mentors. There are lots of mentors for you. I'm not sure that I've played that much of a role, really, but you've certainly been an important part of my life, which you came into when I was setting up my lab for the first time at what is now Imperial College, one of the campuses there. And it was an exciting time, new lab, new money, just getting some new people in. As my chance, I came down from Oxford with Jane, who was sitting over there, who was really my first postdoc. But Rafa was the first postdoc I appointed in my new position. And I vaguely remember the interview. I do remember being really impressed with the fact you've got this paper out where you sequenced and analysed the African swine fever virus genome. That was a big deal in this. Rafa's explained it all to you. It was a big deal in those days. I'd done 8 kb, he'd done 200. So I was really impressed with that. I didn't do 8 kb of the virus, it was something else. I also got a phone call from somebody who'd been working with Jeff Smith in Oxford. I can't remember which stage of your career that was. It was a sort of build-up to doing your postdoc. And he said, yeah, I'd like Rafa in my lab if I could have him. So I thought, right, we'll get this guy. And I never regretted it. Rafa was a great colleague. I mean, so many qualities. His tenacity, his ability to assimilate knowledge in so many different parts of the work. You know, he knows about so many viruses. He knows about so many techniques. He knows the life cycles of all these viruses. He knew that before he even came. and just his ability to communicate, do the work properly and solidly, and write it up. And my only regret is that, as he mentioned, it didn't really come to as much as we'd hoped it would come to. We didn't find the all-important way of tweaking cells so that they would correct the gene in the way we wanted to correct them. And like a lot of these scientific projects, as Rafa mentioned, somebody else comes in and does it very easily much later on. Well, maybe not that easily. So now that's all done with CRISPRs. Everyone's heard of CRISPRs, I think. That was the trick that we didn't get. But on the way, Rafa did some great work, published some very useful papers. They weren't major Nature Genetics papers, but they were good papers and something to be proud of. And we learned a little bit about the whole process. So, and on top of that, it's just, he became a friend. I mean, I remember having lots of talks with Rafa about various subjects, politics and what have you, religion and all these kinds of, his service, his national service and all this kind of thing. It was also an exciting time because Jane and Rafa and I, there must have been something in the water in the lab there. We all became parents about the same time. So we had a lot of other experiments to keep notes on and compare notes on. I wouldn't say it was a collaboration, but... So, thank you, Rafa, for everything today and back then. And I'm sure... So the second thing I need to do is to thank everybody in the audience. I know a few faces, but only a minority, and I'm sure you've come from far and wide, and it's a testament to Rafa's career and his colleagues that everyone has turned up and enjoyed your talk. And then finally, I want to thank the organisers for putting us on, the Royal Holloway and the Department of Life Sciences. I'm quite impressed with the fact they showed us around the campus beforehand. We saw the beautiful... It's always impressive coming down to this campus. Rafa and I worked together for 6 years in not the prettiest campus, next to Wormwood Scrubs prison in West London, and I'm still there. So have pity on me. No, it's all right. But it is very pleasant to come down and see this leafy campus. And I think we should probably, at Imperial College, when people give their inaugural lectures, make sure that the audience gets shown around the V&A Museum, which is right next door there. See if I can work on that one. So, what else to say? I mean, you know, not only was... I mean, I didn't follow every step of Rafa's career after that, but obviously I kept an eye on it. And, you know, I saw him going from strength to strength, using all his qualities you saw his qualities of communication tonight. I mean, that was just one of his many qualities. And building up this work, his editorial work for gene therapy, the rare disease outreach work he's done, the British Society for Gene and Cell Therapy. And yes, maybe you did take on too much, but you're back in business. And I just want to wish you well, keep going, and go on to higher things. Rafa, congratulations. Thank you very much.
Well, Andy, thank you again also for that very, very generous and heartfelt vote of thanks. So, ladies and gentlemen, I think this sort of concludes the formal part of proceedings. But again, I think you've really had a real treat, and it's really gives me enormous pleasure, indeed pride actually, to be Dean of this school. And I think Rafael has really demonstrated all the wonderful qualities that I think our very best colleagues have. And I think your honesty, humility, as I said a moment ago, is just... It's very, very inspiring. Anyway, the good news is we have a reception for you outside there. You've earned your refreshment. But before we do, I really would like to urge us to give one more vote of thanks to a really splendid performance tonight. Thank you again, Rafael.
So just to finish, thank you, and we do have a full buffet for you, so please don't be shy, and join us, and let's make that there is no food left over, and maybe even no drink either. So please join us outside for a celebration, and thank you for coming.
Really, really.